Imagine you are playing the highest-stakes game of hide-and-seek imaginable. You're searching for a dangerous intruder, but right before you kick down the door and find them, they change their internal chemistry to become completely invisible.
Yeah, you could be staring right at them and see absolutely nothing. It is a terrifying thought, honestly.
Right. But on a microscopic level, this is a literal life-or-death battle that is happening inside the human body right now. It's like the ultimate survival tactic.
It really is. And unpacking exactly how that works is our mission for today's deep dive.
Exactly. We are exploring how cancer cells pull off this biological disappearing act. And our source material today is it's a fascinating preview article from the 2026 journal Cancer Cell. It's by researchers Galuzzi, Manek, and Vitale.
Right, and they're basically breaking down a truly groundbreaking study by Foydart and a team of scientists who actually mapped this entire process out.
Yeah, and to really appreciate what these scientists discovered, I think we first have to understand the baseline of the battlefield, right? Like, for a tumor to survive, grow, and actually make you sick, it has to overcome a massive hurdle.
Oh, absolutely. It must evade the body's primary assassins. We're talking about the CD8-positive cytotoxic T lymphocytes.
The CTL.
Exactly. Think of these as like elite patrolling immune cells that constantly scan your tissues. They're just looking for any abnormal cells that threaten to disrupt the body's balance.
Right. So if the immune system is that vigilant, the cancer has to get creative. And the researchers outlined a really helpful framework for this, calling it the 3 C's of immune evasion.
Right, the 3 C's.
So first, there's camouflage, which is simply hiding from the immune cells. Second is coercion, where the cancer actively manipulates or disarms the immune cells that get too close.
Yeah, it fights back.
Yeah. And third is cytoprotection, basically building up physical or chemical shields to survive the attack once the immune cells actually start firing.
It's an incredibly clear way to view tumor progression. But today, our lens is focused entirely on that first C, camouflage.
Just the hiding part.
Exactly. We are looking specifically at a chaotic, massive cellular event called whole-genome doubling, or WGD for short, and we'll see how it gives cancer cells the power to basically rewrite their own visibility.
Okay, well, before we get into doubling genomes, let's talk about normal visibility. If I'm a healthy cell, or even a newly mutated cancer cell, how does the immune system know I'm there? How do I broadcast my identity?
Right. So every cell in your body uses a system called the antigen processing and presentation machinery. Let's think of it as a microscopic flagpole extending from the surface of the cell.
Okay, a flagpole.
Yeah. The pole itself is made of something called an MHC class I molecule. But, a flagpole that tall and heavy, it needs a sturdy base to keep it anchored right.
Makes sense.
That crucial anchor is a protein called beta-2 microglobulin, or B2M.
So you have the B2M base and the MHCI pole sticking out of the cell. What's the actual flag?
Well, the cell is constantly chopping up little bits of its own internal proteins and running them up that flagpole. Those little bits are the flags.
Got it.
So when those elite T cells float by, they inspect the flag. If the flag is made of normal, healthy protein, the T cell just, moves along.
But if the flag looks abnormal, like a mutated cancer protein, the T cell locks on and attacks.
Precisely.
Which means if the cancer cell wants to survive, the most logical thing to do is just take down the flagpole. I mean, if there's no MHCI pole or B2M base, the T cell literally has nothing to inspect.
Exactly. If they dismantle that machinery, the immune system cannot see them at all. But dismantling foundational cellular machinery isn't just some small tweak. It requires a massive biological upheaval.
And that is where whole genome doubling comes in. The researchers focused heavily on breast cancer for this, right? Noting that WGD is this massive, often underestimated source of tumors with wildly abnormal chromosomes.
Yeah, aneuploid tumors.
Right, and I want to make sure I understand the mechanics of this. From what I read, 2 cells can literally fuse together. They merge their contents, and suddenly, this single supercell has double the genetic material.
Yes. They use specific fusogenic mediators to basically blend their membranes. It is a completely catastrophic event for a normal cell.
Let me try an analogy to visualize this for you because it helped me. Imagine a highly efficient factory, and one day, the machinery glitches and blindly photocopies its entire master genetic blueprint. Every single page just doubled.
It's pure chaos.
Total chaos.
Yeah.
Normally, a factory like that has a strict quality inspector.
Right.
In our cells, that inspector is a famous protein called P53. If P53 sees a cell blindly doubling its entire genome, it's supposed to hit the emergency stop button and force the cell to self-destruct.
That is a brilliant way to frame it. And it sets up what is arguably the most fascinating paradox in this entire field of research.
The p53 paradox.
Yeah. When the scientists looked across huge data sets of cancer genomes, they found that whole genome doubling happens most frequently in tumors, where that p53 inspector is completely broken or mutated.
Well, yeah, that makes intuitive sense. I mean, if the quality inspector is asleep at the wheel, the factory gets away with the crazy photocopying error.
It does make sense. But here's the paradox. The most aggressive, metastatic, poor prognosis breast cancers are actually the ones where the genome doubled, but the p53 inspector was intact and fully awake.
Wait, hold on. Why would the cancer be more dangerous if the body's strict quality inspector is still active? Shouldn't an active p53 be a good thing?
You would think so, right? But it comes down to extreme evolutionary pressure. If a cancer cell undergoes this chaotic genome doubling and the p53 inspector is wide awake, that cell should die instantly.
Right.
If it manages to survive anyway, it means that specific cell has somehow adapted to bypass the absolute strictest security system the human body has to offer.
Oh, wow.
Yeah, surviving whole genome doubling with an intact p53, or TP53 competent, as they call it, represents a highly selected, elite evolutionary state. These aren't just surviving cells, they are the ultimate apex predators of the microscopic world.
That is wild. So to study these apex predators without the confusing variable of a broken inspector, the researchers engineer their own matched cells. They force breast cancer cells to fuse, doubling their genomes, but deliberately ensured their p53 responses remained perfectly intact.
Exactly. They wanted to isolate the specific advantages of whole genome doubling.
Okay, let me pause here and stitch this together for you, because this is a really wild foundation. So let's do a quick 5-minute mini recap of our first major segment.
Good idea.
We started with the 3 C's of immune evasion: camouflage, coercion, and cytoprotection. We learned that to achieve camouflage, a cell needs to dismantle its MHCI flagpole and its B2M anchor. Then, we look at how cells pull this off through a chaotic doubling of their DNA, leading to this crazy paradox. The cancer cells that survive our body's strictest p53 quality control actually evolve into the most elite, dangerous threats.
Perfectly summarized.
So think about that for a second. Have you ever considered that the very systems designed to protect us can inadvertently forge a much stronger enemy if that enemy manages to survive the initial test? What do you think happens to a cell's behavior once it successfully doubles absolutely everything inside itself?
Well, it's natural to assume that if a cell doubles its blueprints and its machinery, it's just going to grow twice as fast.
That would definitely be my assumption. More blueprints equals more production equals faster tumor growth.
It's a common myth in oncology, honestly, but it's fundamentally incorrect. The researchers proved that in a petri dish, these WGD-positive clones do not proliferate faster. They don't have some magical growth advantage.
Really, if they aren't growing faster, what is the point of lugging around double the genetic baggage?
It grants them something called phenotypic plasticity.
Phenotypic plasticity.
Right. They physically get larger, yes, but more importantly, they completely remodel how they transcribe their genes. They become incredibly adaptable. Instead of focusing on dividing rapidly, they focus on becoming flexible survivors, creating massive variety within the tumor.
But surely that flexibility comes with a hidden cost. I mean, doubling millions of base pairs of DNA has to be a logistical nightmare for this cell.
Oh, it is absolute chaos. And that chaos creates a major vulnerability. The study highlights a concept called non-oncogene addiction.
Right, non-oncogene addiction, because usually we think of cancers being addicted to mutated cancer-causing genes.
Exactly. But in this doubled state, the cells become desperately dependent on a completely normal, healthy gene just to keep from falling apart.
I find this fascinating. What is the normal gene they become addicted to?
It produces a protein called BIRC5. Because these cells have a massive, unwieldy double genome, physically dividing into 2 cells without shredding their DNA is incredibly difficult.
That makes sense.
BIRC5 is a master regulator of mitosis and apoptosis, basically cell division and programmed cell death. Without it, the doubled cell simply cannot manage the logistics of division. If you use a drug to block BIRC5, these elite cancer cells die. It is their ultimate Achilles heel.
Okay, wait. If scientists have identified this glaring BRCA5 weakness, why are these tumors still surviving and thriving inside an actual living patient?
Because a petri dish is very different from the human body. To explain how they survive in a patient, the researchers detailed an epic 3-part evolutionary war. Feudart's Figure 1 outlines this perfectly. It happens in 3 distinct phases. priming, immunoediting, and escape.
Let's walk through that battlefield. Phase 1 is priming. What kicks this off?
Remember that carrying a double genome is stressful. Specifically, it causes immense stress in a part of the cell called the endoplasmic reticulum, or ER.
Right.
Because of this ER stress, the cancer cell accidentally pushes a specific protein to its outer surface. That protein is called calreticulin, or CALR.
And from my understanding, calreticulin acts like a massive flashing neon sign that says, "Eat me."
That is exactly what it does. It is a major distress signal. Specialized immune cells, known as dendritic cells, constantly patrol the tissues looking for that exact signal using something called the LRP1 receptor.
Right.
When they see the calreticulin via LRP1, the dendritic cells literally gobble up the stressed, doubled cancer cells.
But if the dendritic cell eats the cancer cell, doesn't that just solve the problem right there? Why is there a phase 2?
Because the dendritic cell isn't the executioner, it's the intelligence officer. It digests the dead cancer cell, takes the most recognizable physical chunks of it, and travels all the way to your lymph nodes.
Oh, wow.
It holds these chunks up to the elite T cells we talked about earlier and essentially says, memorize this shape, go find anything in the body that looks like this and destroy it. This is a process called cross-priming.
Which brings us to phase 2, immunoediting. The intelligence has been passed on, and the assassins are deployed.
The battlefield erupts. The CD8 positive T cells flood into the tumor environment. They are actively hunting for those MHCI flagpoles flying the cancer flags.
I want to know exactly how they take the cancer down. I mean, they don't just bump into them, right? How does a T cell physically kill the target?
It's a highly coordinated chemical attack. When the T cell locks onto the flagpole, it releases specialized proteins. The first protein is called perforin 1, or PRF1. It acts like a microscopic drill, physically punching a hole right through the cancer cell's outer membrane.
A microscopic drill. That is intense.
Yeah. Then it drops a second protein directly into that newly drilled hole. That second protein is granzyme B, or GZMB, which is essentially a chemical grenade that triggers an unstoppable self-destruct sequence inside the cancer cell.
That is brutal. So the immune system is actively slaughtering the tumor. But you called this phase immunoediting. That implies the immune system is somehow editing or shaping the cancer.
It is Darwinian evolution playing out in real time. The immune system is applying immense chemical pressure, specifically through an inflammatory signal called interferon gamma, or IFNG.
Okay.
The T cells act as a massive sieve, slaughtering every single cancer cell that is visible.
Which means the only cells that survive the massacre are the mutants, the anomalies.
Exactly. This is phase 3, escape. Under this brutal, relentless IFNG pressure, a tiny fraction of the doubled cancer cells manage to completely shut down their antigen processing and presentation machinery. They dismantle the flagpoles, the immune system is still swarming the area, weapons drawn, but suddenly the targets they vanish.
And the researchers actually watched this happen over time, right? They saw the tumor environment completely shift. It started out immune swarming with attacking T cells and evolved into an immune cold environment.
Yes.
The cancer cells even stopped sending out the chemical distress beacons like CXCL9 and CXCL10 that normally guide T cells to the tumor site.
Yeah, they go totally dark. And to definitively prove that taking down the flagpole was the master mechanism, the scientists ran a brilliant control experiment.
Right, the B2M experiment.
Exactly. They took regular, non-doubled cancer cells and artificially deleted the gene for the B to M anchor, the base of the flagpole. Those regular cells immediately achieved the exact same invisibility as the elite doubled tumors. It proved that dismantling the flagpole is the absolute core of their camouflage.
So the narrative here is wild. Let's do another 5-minute mini recap of this second segment, summarizing this crazy battlefield.
Let's do it.
First, we realize that doubling a genome doesn't make cells grow faster. It makes them adaptable and plastic. Second, this chaotic doubling saddles them with a desperate addiction to the BRUC5 protein just to manage their own cell division.
Right.
And third, they survive a brutal 3-stage war. They get stressed and eaten by dendritic cells, which prime the T cells to attack with microscopic drills and grenades. Finally, the only survivors are the ones that manage to pull down their flagpoles, turning the whole area immune cold.
That's the perfect summary.
So if you were a scientist looking at this incredible sequence of events, where would you strike? Would you try to design a drug targeting that hidden BRC5 weakness? Or would you try to figure out a way to strip away the tumor's newly acquired camouflage?
Well, the team behind this research was heavily invested in the latter. They didn't just want to know that the cells pulled down the flagpoles. They wanted the exact granular molecular blueprint of how they did it, the 4-lane route to invisibility.
Right. It's easy to say they turn off the genes. It's an entirely different level of science to trace the exact biochemical wires they snip to make it happen.
Oh, absolutely.
And the paper describes this as a highly specific epigenetic pathway. To help us track this complex chain reaction, I want to use a simple mnemonic that I came up with, SHH.
Right.
Like telling someone to be quiet, which is essentially what the cancer is doing to its own DNA. SHH.
I love that mnemonic. It perfectly maps to what's happening. Let's break down the SHH mechanism. The first letter S stands for succinate.
Succinate.
Right. Succinate is a completely normal, naturally occurring byproduct of everyday cellular metabolism.
It's just molecular exhaust, basically.
Exactly. But in these double cancer cells, their metabolic wiring gets warped. They can't process the exhaust properly, and succinate begins to heavily accumulate inside the cell, filling it up like water in a sinking ship.
Okay, so the cell is flooding with succinate. What is that physical buildup damage? Because that brings us to the first H.
The first H stands for Holt's KDM6.
Holt's KDM6.
To understand this, we need a really quick look at epigenetics. Your DNA isn't just a loose string floating around. It is tightly wrapped around chemical spools called histones. You have specialized enzymes that act like writers. They add chemical tags to these spools that say, do not read this gene.
Right.
And you have enzymes that act like erasers. Their entire job is to remove those tags so the gene can be read and built. KDM6, which is a lysine demethylase, is one of those erasers. Its specific job is to remove the do not read tags off the genes that build the MHCI flagpole.
But the succinate gets in the way.
Exactly. So succinate is a potent inhibitor of KDM6. It paralyzes it. Succinate halts KDM6. The eraser is effectively broken.
Which leads us perfectly to the final H3K27E3 buildup. Now, that's a massive mouthful of letters and numbers, but it's just the scientific name for that specific do not read tag.
Right.
Because the succinate broke the eraser can't do its job, so these repressive tags just start to build up uncontrollably.
Exactly. SHH, succinate halts KDM6, leading to H3K27E3 buildup. Those tags clamp down incredibly tightly on the DNA. The instructions for building the MHCI pole and the B2M anchor are completely buried. Production stops. The flagpoles are dismantled. Camouflage is achieved.
That is a stunning chain reaction.
Yeah.
The cell is literally using a shift in its metabolism to alter its epigenetics, creating total immune invisibility. But when I was reading this, there was a tiny, brilliant twist regarding the enzyme that actually writes those tags.
Yes, this is a crucial nuance. The enzyme responsible for writing that specific do not read tag is called PRC2. Now, if you look at a cell and see a massive pileup of these tags, your first instinct would be that the PRC2 writer has gone rogue, right? That it's working overtime, frantically stamping tags everywhere.
That's what I assumed. If there's too much ink on the page, you normally blame the pen.
But that's not what's happening. The PRC2 writer is working at a completely normal baseline pace. The heavy accumulation of tags is entirely due to the broken eraser. The writer is just doing its normal job, but because no one is cleaning up behind it, the ink just piles up until the genetic instructions are totally buried.
But here's the therapeutic miracle that just blew my mind. Even though the PRC2 writer isn't the part of the system that actually broke, using a pharmacological drug to block the writer still acts as a cure.
It does. It's amazing. If you give these invisible tumors a PRC2 inhibitor, you paralyze the writer. Since the eraser is broken, paralyzing the writer stops any new tags from being added. Over time, the heavy accumulation of tags naturally fades away. The epigenetic suppression lifts.
It's like turning on a high-powered flashlight in a pitch-black room.
Yes.
Suddenly, the genes wake up.
Yes. The researchers observed that when you inhibit PRC2, the signaling pathways for interferon gamma, specifically effectors like JGA2 and STAT1, roar back to life.
Why?
The cell immediately begins rebuilding the antigen processing and presentation machinery. The MHCI flagpoles go back up, the flags are hoisted, the tumor becomes completely visible to the immune system once again, and those elite T cells swarm back in to suppress the tumor.
Man, that is just, it's so elegant. Let's do our final 5-minute mini recap here to consolidate this incredible masterclass in cellular biology.
Sounds good.
We track the SHH pathway. A shift in metabolism causes succinate to accumulate. That exhaust halts a crucial eraser enzyme called KDM6. Because the eraser is broken, repressive chemical tags called H3K27Me3 build up, completely burying the genes needed for visibility.
Right.
But by deploying drugs that inhibit the PRC2 writer enzyme, Scientists can lift the suppression, force the flagpoles back up, and strip away the cancer's invisibility cloak.
Spot on.
Think about the sheer elegance of that. Isn't it mind-blowing that a cancer cell utilizes a basic, everyday byproduct of cellular metabolism, literally just exhaust, as the master switch to rewrite its entire genetic destiny and hide from the immune system?
It really is. And, if we zoom out and look at the broader implications of this paper, the potential is staggering. We know that the vast majority of clinically dangerous tumors, the ones that ultimately threaten a patient's life, are incredibly messy and have abnormal chromosomes.
Right.
This research suggests that a huge portion of those deadly tumors utilize this exact intermediate whole genome doubled state as a bridge to acquire their invisibility.
So they use the chaos of doubling as a deliberate stepping stone.
Precisely. Foydart and the team have delineated a foundational mechanism here. They explained exactly how highly abnormal cells, which should be easily spotted and destroyed, manage to establish a hidden reservoir for deadly cancer progression. By illuminating this pathway, they have handed the medical community a map. Epigenetic modulators, like PRC2 inhibitors, represent a highly promising tool to artificially strip away this camouflage in the clinic.
The researchers do note, of course, that cancer is always evolving, right?
Yeah.
Whether these specific inhibitors will work on older, deeply entrenched tumors that have mutated far beyond their initial doubled state is something that, well, it requires a lot more formal investigation.
Right. That is the nature of science. Every answered question uncovers 3 new ones. But the foundation laid by this study is a massive leap forward in understanding tumor evolution.
It really is. And it leaves me with one final lingering thought for you to chew on as we wrap up. We just spent this entire deep dive mapping out how cancer cells accumulate succinate, a totally natural, foundational metabolic byproduct to trigger this cascade of invisibility. It makes you wonder, if these cells are literally hijacking the foundational components of cellular metabolism to hide, could our body's broader everyday metabolic state ultimately influence how well these camouflage tactics actually work? Are we, through our own internal chemistry, inadvertently providing the raw materials for their invisibility cloak?
That is a phenomenal question, and it really speaks to the growing, vital intersection of lifestyle, systemic metabolism, and oncology.
Thank you for joining us in the deep end today. Keep asking questions, keep looking beneath the surface, and remember that even the most perfectly invisible threats leave a chemical trail if you know exactly how to look for the missing flags.